Hi, hello viewers. Welcome back again to one more other lecture session from this channel. So in this lecture session, we will be studying regarding the structure of DNA. So in our previous lecture session, we have studied regarding the structure of the polynucleotide chain. And in this lecture session, we will be studying regarding the structure of DNA, which was elucidated by James Watson and as well as Francis Crick in detail. So here, prior to the identification of the DNA from the nucleus of the cell, here, the acidic content which is present in the nucleus of the cell for the very first time, it was identified and it was observed by Friedrich Miescher and he named it as nuclein. So here, prior to the identification of the DNA from the nucleus of the cell, here the acidic content which is present in the nucleus of the cell, for the very first time it was identified and it was observed by Friedrich Miescher and he named it as nuclein. So just let me write it here. The acidic content present in the nucleus of the cell was identified and observed by Friedrich Miescher and named it as nuclein. And here, because of the technical limitations in those days, successful isolation of the polynucleotide chain from the nucleus of the cell, it was not took place. And however, because of the collaborative work which was carried out by James Watson and as well as Francis Crick, by taking the advantage of the X-ray diffraction data which was provided by Maurice Wilkins and as well as Rosalind Franklin, these people, they have explained the structure of the DNA, which is very simple but became so famous. So just let me write it here. Because of technical limitations, isolation of the polynucleotide chain, from the nucleus of the cell, was not took place. And here, because of the collaborative work which was carried out by James Watson and as well as Francis Crick, by taking the advantage of the X-ray diffraction data which was provided by Maurice Wilkins and as well as Rosalind Franklin, these people, they have explained the structure of the DNA. James Watson and Francis Crick, by taking the advantage of X-ray diffraction data provided by Maurice Wilkins and Rosalind Franklin, they have explained the structure of the DNA. They have explained the double helical structure of the DNA. So here the double helical structure of the DNA, which was illustrated by James Watson and as well as Francis Crick, though it is very simple, but it becomes so famous. And here one of the hallmark property of the double helical structure, which was explained by James Watson and as well as Francis Crick, here it is nothing but what? base pairing in between the nitrogenous bases. So here certain nitrogenous bases like adenine, guanine, thymine and as well as cytosine in general, they exist in case of the double-stranded DNA. And here all the nitrogenous bases, they are capable to undergo base pairing because of the complementary relationship. So just let me write it here. All the nitrogenous bases like adenine, guanine, thymine, cytosine are capable to undergo base pairing because of complementary relationship. So here always the concentration of purines, it should remain equal with the concentration of pyrimidines. And here for the very first time it was explained by Erwin Chargaff. So here Erwin Chargaff, a famous biochemist, for the very first time he explained that in case of one particular double-stranded DNA, the concentration of purines and as well as concentration of pyrimidines, it should remain similar or it should remain same. That means in case of one particular double-stranded DNA, if the concentration of purines is, if it is around 50%, under such condition the concentration of pyrimidines, it should remain 50%. And here the specific strategy, it was known as Chargaff's rule. So according to the Chargaff's rule, which was explained by Erwin Chargaff, the concentration of purines and as well as the concentration of pyrimidines, it should remain similar or it should remain same. So here the concentration of Erwin Chargaff for the very first time explained that in double-stranded DNA, the concentration of purines and pyrimidines should remain similar. And here the particular strategy, it came to know as Chargaff's rule. Is it clear? And then after we will be studying regarding some of the salient features of the double-stranded DNA, which was explained by James Watson and as well as Francis Crick in detail. So let me arrange the things here. And here, as already mentioned, all the nitrogenous bases which do exist in case of the double-stranded DNA, they are capable to undergo base pairing. It is because of what? It is because of the complementarity relationship. So here, because of the complementarity relationship in between the nitrogenous bases, so here all the nitrogenous bases, they are capable to undergo base pairing. All the nitrogenous bases are capable to undergo base pairing because of complementary relationship. Complementary relationship, right? So here, because of the complementary relationship in between the nitrogenous bases, so here by taking the advantage of the nucleotide sequence in one DNA strand, here we can predict the nucleotide sequence in another strand. because of the complementarity relationship, because of the complementarity relationship, by taking the advantage of the nucleotide sequence in one DNA strand, by taking the advantage of the nucleotide sequence in one DNA strand, we can predict the nucleotide sequence in another strand. The nucleotide sequence in another strand. Right? So here, in order to understand this particular concept, here we can take the advantage of one of the common example. So here let us assume one particular double-stranded DNA. So it is in 5' to 3' end, and here it is 3' to 5' end. Is it clear? So initially we don't know the nucleotide sequence in this particular DNA strand. So here, by taking the advantage of the nucleotide sequence in this particular DNA strand, which is in 5' to 3' polarity, here we can predict the nucleotide sequence in the another DNA strand, which is in 3' to 5' polarity. So here let us assume some of the nucleotides like AT, GC and AT. So here, by taking the advantage of the nucleotide sequence in this particular DNA strand, here we can predict the nucleotide sequence in another DNA strand. So here the complementarity of A is, obviously it should be thymine, and the complementarity of T is A, complementarity of G is C, complementarity of C is G, complementarity of A is T, and the complementarity of T is A. So initially we don't know the nucleotide sequence in this particular DNA strand, but here, by taking the advantage of the nucleotide sequence in one particular DNA strand, here we can predict the nucleotide sequence in another DNA strand. It is because of what? Because of the complementarity relationship. Is it clear? And here both the strands of the DNA, they act as template in order to regulate the replication mechanism. So here, during the replication mechanism, from one particular parental double-stranded DNA, a daughter double-stranded DNA, generally it synthesizes here. So here, in order to synthesize one particular daughter double-stranded DNA from the parental double-stranded DNA, both the single-stranded DNAs, they act as template in order to regulate the replication mechanism. Both the strands in double-stranded DNA, act as template in order to regulate the replication mechanism. And here, because of these salient features, the genetic implications of the particular DNA or the deoxyribose nucleic acid, it had become very clear. Is it clear? Because of all these salient features, the genetic implications of the deoxyribose nucleic acid, it had become very clear. And here, right now, we will be studying regarding the various salient features of the double helical structure, which was explained by James Watson and as well as Francis Crick in detail. So let me erase the things here. And here, if we consider some of the salient features which are in association with the double helical structure, which was explained by James Watson and as well as Francis Crick. So here, basically DNA is double-stranded. That means it is having two single strands here. So here, one DNA strand, it proceeds in 5' to 3' polarity, and whereas the other DNA strand, it proceeds in 3' to 5' polarity. Both the strands in DNA, has anti-parallel polarity. Anti-parallel polarity. And it may include 5' to 3' polarity, and the other one is 3' to 5' polarity. And here each of the particular DNA strand, it is having many of the nitrogenous bases here. So here all the nitrogenous bases, they are capable to undergo base pairing because of the complementarity relationship. And in addition to the complementarity relationship, in order to provide the stability in between the two single strands here, there is a requirement of a specific non-covalent interaction, which is nothing but what? Hydrogen bond. Hydrogen bond provides the stability in between two single strands here. Two single strands. So here the strongest hydrogen bonding in between the two successive nitrogenous bases, it occurs in between G and C and C and G. G and C and C and G. So in between G and C and C and G, there is an availability of 3 hydrogen bonds. So it is the strongest hydrogen bond. And in comparison with G and C and C and G, the weaker hydrogen bonding, it occurs in between A and T and T and A. A and T and T and A. So in between A and T and T and A, there is an availability of only 2 hydrogen bonds. So it is a weaker hydrogen bond. And in comparison with A and T and T and A, the much more weaker hydrogen bonding, it occurs in between A and U and U and A in case of RNA. But whereas in case of DNA, the strongest hydrogen bonding, it occurs in between G and C and C and G, and the weaker hydrogen bonding, it occurs in between A and T and T and A. And in addition to this, here we need to understand some of the topological features which are in association with the double-stranded DNA. And here these may include the pitch of the helix. What it is? Pitch of the helix. Pitch of the helix, or it is otherwise known as one helical turn. One helical turn. So here the pitch of the helix or one helical turn, it may range around 3.4 nanometers, or in other terms we can consider it as 34 angstrom units. 3.4 nanometers or 34 angstrom units. So always try to memorize one thing. 1 nanometer, it is equal to 10 angstrom units. 1 nanometer, it is equal to 10 angstrom units. So in that case, 3.4 nanometers, it is equal to 34 angstrom units. And the distance between the two successive nitrogenous base pair, it ranges around 0.34 nanometers. 0.34 nanometers. So let me write it here. Distance between two successive nitrogenous base pair. Two successive nitrogenous base pair. It ranges around 0.34 nanometers, or in other terms we can consider it as 3.4 angstrom units. So here, in order to calculate the number of base pairs which are present in case of one helical turn, here we need to multiply 0.34 nanometers into 10. So under such condition, we obtain here 3.4 nanometers or 34 angstrom units. Because the distance between the two successive nitrogenous base pair, it ranges around 0.34 nanometers. And as already mentioned, in case of one helical turn, there is an availability of 10 base pairs. So once when we multiply 0.34 nanometers into 10, here we obtain 3.4 nanometers or 34 angstrom units. And in addition to this, when the particular DNA strand, when it proceeds from one particular base pair to another base pair, the DNA strand, it turns around 36 degrees. DNA strand turns around 36 degree when it proceeds from one base pair to another base pair, from one base pair to another base pair. So here all the nitrogenous base pair, they stack upon another nitrogenous base pair. So here, because of the presence of the nitrogenous base pair in very close proximity, and in addition to this, because of the presence of non-covalent interactions like hydrogen bond, both the strands in DNA, they acquire the stability here. Both the strands in DNA, acquire stability because of hydrogen bond. So here the specific nitrogenous bases which are present in case of the double-stranded DNA, they may include purines and as well as pyrimidines. So here purines are double ring structure, but whereas pyrimidines are single ring structure. So here, though there is a variation in case of the ring structures in case of the double-stranded DNA, but because of the presence of a non-covalent interaction like hydrogen bond, both the strands of the DNA, they acquire the stability. So here, after the successful elucidation of the DNA double helical structure by James Watson and as well as Francis Crick. So here, though the structure of the DNA, though it is very simple, but here the genetic implications which were induced by the double helical structure, it had created a revolution here. So here, after the successful elucidation of the DNA double helical structure by James Watson and as well as Francis Crick, within no time, Francis Crick, he had explained the central dogma of molecular biology. So here, according to the central dogma of molecular biology, each and every gene which is a segment of DNA, when it participates in gene expression, it synthesizes in single-stranded mRNA. And here, when the specific mRNA, when it participates in case of the translation, it is capable to regulate the synthesis of protein. So just let me show it here. Each and every gene, when it undergoes expression during the transcription mechanism, it synthesizes mRNA. And mRNA, when it participates in case of the translation, it synthesizes protein. Is it clear? So here it is termed as what? The central dogma of molecular biology. Central dogma of molecular biology. So for the very first time it was explained by Francis Crick. And in addition to this, here we need to understand one more other alternative strategy, which is nothing but what? Reverse transcription. So here, what is reverse transcription here? So here, in order to understand the reverse transcription, at first we should have a basic idea regarding the transcription mechanism. So in general, during the transcription mechanism, from one particular gene, mRNA is synthesized here. But whereas in case of the reverse transcription, as the name itself suggests, it is the opposite to the transcription. So in case of the transcription mechanism, from one particular gene, mRNA is synthesized. But whereas in case of the reverse transcription, from mRNA, DNA is synthesized. And here it is termed as what? Reverse transcription. In order to regulate the reverse transcription, there is a requirement of a specific enzyme which is nothing but what? Reverse transcriptase. So just let me arrange the things here. Reverse transcriptase. From mRNA, if DNA is synthesized, so here it is termed as what? Reverse transcription. And here, in order to regulate the reverse transcription, there is a requirement of a specific enzyme which is nothing but reverse transcriptase. Reverse transcriptase. So here the reverse transcriptase, it is having a specific enzymatic activity which is nothing but RNA dependent DNA polymerase activity. RNA dependent DNA polymerase activity. DNA polymerase activity. So in general, during the transcription mechanism, in order to synthesize the mRNA from the particular gene, the specific enzyme which is required, here it is nothing but what? DNA dependent RNA polymerase. That means by taking the advantage of the biological information which is present in case of DNA, here mRNA is synthesized. So the reason why here it is termed as DNA dependent RNA polymerase. But whereas in case of the reverse transcription mechanism, the particular reverse transcriptase, it is having RNA dependent DNA polymerase activity. That means by taking the advantage of the information which is present in case of the mRNA, here DNA is synthesized here. So the reason why here it is termed as RNA dependent DNA polymerase, which is termed as the reverse transcriptase. So here most commonly the particular reverse transcriptase, it is observed in case of certain retroviruses like HIV virus. Is it clear? So here this is regarding the double helical structure DNA, which was explained by James Watson and as well as Francis Crick in detail. And in the next upcoming lecture session, we will be studying regarding the packaging of the helix. Is it clear? So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.